Happy Friday, everyone. We just have a real short lesson today to kind of wrap up what we've been learning about energy and specifically photosynthesis. Yesterday, we talked about the details of how photosynthesis works. Today, we're going to talk about some modifications to that process that have evolved in some types of plants to help them survive better and still be able to photosynthesize in different types of environmental conditions. As you know, water is one of the reactants that's necessary for photosynthesis. So especially in places that are hot or dry, plants need to come up with some different adaptations to deal with a lower amount of water or the fact that they keep their stomata closed more often to prevent water loss in those dry environments. So we're going to watch this short video. In your note packet as you watch this, you are going to learn about two different ways that plants have adapted to this. One of them is called C4 photosynthesis and one is called CAM photosynthesis. What I want you to note is just the main difference and how this adaptation helps them to survive in those climates, and maybe just jot down one or two examples of a type of plant that falls into that group. So add some notes to your note packet as we watch this short video.
Photosynthesis is the process by which plants capture the energy of the sun to make carbohydrates. But there are minor variations in how plants do this. Photosynthesis is the process by which plants capture the energy of the sun to make carbohydrates. But there are minor variations in how plants do this, depending on the environment in which the plant lives. In this tutorial, we'll look at the differences between the three types of photosynthesis, C3, C4, and CAM. Most plants, around 85% or so, carry out C3 photosynthesis. As we will see, the name C3 has to do with the type of carbon molecule produced during the process. C3 photosynthesis occurs in the mesophyll cells of the C3 plant, which are located just under the surface of the leaves and other tissues. Let's take a quick look at how photosynthesis occurs in these cells. During C3 photosynthesis, CO2 is taken up and transported directly to the Kelvin cycle, where it's fixed into a 3-carbon molecule, thus the name C3. If the stomata are open, CO2 moves in and oxygen moves out of the cell. But under hot or dry conditions, when the stomata are closed, oxygen accumulates, which inhibits the amount of C3 that can be made. Some plants have evolved an adaptation that allows them to be successful in hot, dry conditions. These plants carry out C4 photosynthesis instead of C3 photosynthesis. During C4 photosynthesis, CO2 moves into the mesophyll cell and is immediately fixed into a 4-carbon molecule, thus the name C4 photosynthesis. But the anatomy of a C4 plant is different from that of a C3 plant. A somewhat simplified version of this process is shown here. In a C4 leaf, chloroplasts are located in mesophyll cells, but they are also located in bundle sheath cells, which surround the leaf vein. CO2 moves from the mesophyll cells into the bundle sheath cells, where the Kelvin cycle occurs. This shields the plant from the oxygen buildup that would otherwise occur when the stomata are closed during dry conditions. A second way of addressing the problem of oxygen is to change the timing of photosynthesis. This is called CAM photosynthesis, and in many ways it's similar to C4 photosynthesis, except that the timing of the process changes. During CAM photosynthesis, CO2 is taken up during the night when the stomata are open and is fixed into a 4-carbon molecule. It's then stored until daylight as an intermediate molecule, which gives the CAM cycle its name. This adaptation allows photosynthesis to occur in drier climates than in both C3 and C4 photosynthesis. A great example of a CAM plant is the cactus. Let's do a quick review. During C3 photosynthesis, CO2 is taken up and transported directly to the Kelvin cycle. The C4 pathway alters the location of photosynthesis and the CAM pathway changes the timing. Both of these adaptations allow some plants to survive in environments that are less than ideal for the typical C3 plant.
Okay. So hopefully you were able to jot down some notes about these different adaptations of kind of how plants have learned to cope or have evolved to cope with those different types of environmental situations. The last thing I want to talk about real briefly is how you need to wrap up your algae bead lab. So the reason that we use algae in this experiment, we could use any type of photosynthetic organism, but algae, because they can do photosynthesis, actually do both of these processes. They make their own carbohydrates and they also do cellular respiration to break them down to make ATP. So when your algae are sitting under light, they are definitely photosynthesizing, but remember, cellular respiration has to happen also because those cells have to continue to make the ATP they need. So as you think about what you were measuring carbon dioxide. Photosynthesis uses carbon dioxide, so it decreases that CO2 level. But at the same time, cellular respiration is producing a little bit of carbon dioxide. So we can never completely just measure photosynthesis because cellular respiration is always happening in there as well. So that's just something to keep in mind as you answer the questions and as you try and figure out exactly what's happening with your data. The last piece, to answer the last 3 questions on your lab document, you will need to discuss your results with some other classmates. If you're not able to join the live session today to do this in person, make sure you reach out to a few other people in class, ask them to share what they did with their experiment and what kinds of results they got. Because the idea here is to use other classmates' experiments, kind of like you would do other trials if we were in the classroom, to draw some broader conclusions about the rate of photosynthesis and respiration that you are measuring. One really important thing, which is also part of those last few questions, is to think about the design of this experiment. Obviously, since we were all in different households, we did some steps the same, and I gave you a basic procedure, but it's really important here to spend some time thinking about what variables could not be controlled. What else might have affected the rate of photosynthesis in this experiment that wasn't necessarily the same in my house and in your house and in your classmate's house? Think about that and make sure that you really write about that as you're answering those last few questions as well, because that's a really important consideration in this particular situation. The Algae Bead Lab is due by Monday. Let me know if you have any questions, and I hope you have a wonderful weekend. I'll see you on Monday.